All right, so as we're going to walk through cellular respiration, I'm going to show you kind of the big picture. First, we're going to start with glycolysis. Lysis. I'll put this in a square, all right? And our location of glycolysis is in the cytosol. So what I'm going to do is I'm going to go ahead and this is going to be my cell, all right? So this is we'll call it our cellular environment. Just meaning the outside of this cell. And this is going to be our cytosol. All right, this little guy here, this is our plasma membrane. And I'm going to draw mitochondria, and the way I'm going to do that is I'm going to have this circle, and this is going to represent our mitochondria. I'm also going to have cristae that I'm going to draw, so these interfoldings are called cristae. All right. And when we're looking at the our various processes, I'm just going to label this quick. We have our matrix, our inner membrane, and the outer membrane. All right, so when we're undergoing the processes of photosynthesis and cellular respiration, just cellular respiration, we are going to have glycolysis occurring here in the cytosol. As you can see here, cytosol, this is where glycolysis is going to occur. I'm going to draw a square indicating where the Krebs cycle is going to occur, and that's in the matrix here. And then we're going to have the electron transport chain, or oxidative phosphorylation, you might hear it called. It's going to occur here in this, in the inner membrane. All right. We'll zoom in on each of these a little bit later, okay? But let's look at glycolysis. All right. When we're undergoing glycolysis, when we're starting out with glucose, and in the end, in the electron transport chain, we want to have electrons that are donated in order to create ATP, right? We want to get ATP in the end. But we need to start generating that energy, those electrons, to pump along the electron transport chain. All right, so step one of that starts in glycolysis. So I'm going to just zoom in here, and we're going to talk about glycolysis, okay? And now, like I said, glycolysis has 10 steps, and we don't need to know all those 10 steps. Here's really what you need to know at this point in your biology careers, is that we're going to start with glucose. So I'm going to draw this 6-carbon molecule, and this is going to represent glucose. All right? And glucose has a lot of energy. It's come from derivatives of the Calvin cycle. It has a lot of energy stored up in it, and we're going to use this energy to donate the energy represented in electrons to NADH and ATP, all right? So in order to do that, I'm going to draw this arrow representing the reaction, but in the end, we want to end up with 2 3-carbon molecules, all right? And these are going to be called pyruvate. And each glucose yields 2 pyruvate. Now, in order to do that, we have to do several things, okay? There's going to be several steps in between here, and it's going to take gaining energy and using energy in order to get glucose to pyruvate. So what does that look like? Well, at some point, we need to use energy in this step, and then a little bit later on, we're going to actually take energy from glucose. So first, we're going to take ATP, which you can see here. We've got our ATP, and we're going to give one of the phosphates to glucose and form ADP. We're going to do that a couple of times, so there's really going to be using 2 ATP. And then for each of these pyruvates, later on, we are going to take NAD+. I don't want that to be blue. I'm going to do green for NAD. NAD+, All right, and we're going to use that NAD plus to carry or snag some of the electrons from glucose to create NADH. All right, we're also going to take more of ADP plus P floating around in the cell, not necessarily this one, but other ADP plus P, and we're going to make more ATP. All right, and these two ATPs and our NADH, remember this part really is happening twice, one per pyruvate. So in this process, we are going to yield 2 ATP. So I'm going to have like a count of ATP going. I'll have that over, we'll keep that over here. I'll put it next to this actually. 2 ATP. I'll tack it on to glycolysis. So we have a gain or a net yield of 2 ATP coming from this process. All right. Okay, there we go. Now we've zoomed out. So we're going back. This is our picture of what we've got so far. We've got glycolysis producing pyruvate. Now, in order to continue the next step of the cycle, this pyruvate is going to be moved into the mitochondria. All right, so it's going to come into the mitochondria via active transport. All right, pyruvate is going to be transported from the cytosol into the mitochondria, where we're going to further oxidize our pyruvate. Okay, we're going to start the Krebs cycle, but remember, to get to the Krebs cycle, we have one teeny step to do. So in our Krebs cycle, our pyruvate, I'm just going to do one pyruvate here, but just remember that ultimately from one glucose we get 2 pyruvate. What's going to happen is this pyruvate needs to be, I want this to be black. Pyruvate is going to be oxidized, where we are going to have an enzyme called coenzyme A bind to pyruvate, and we are going to end up with acetyl-CoA plus pyruvate, which is called acetyl-CoA. And as a result of this, we are also going to lose a little bit of carbon dioxide. This is where oxidation occurs, where we are losing some electrons to carbon dioxide. That carbon dioxide is going to leave the cell. Now, something else that we're going to do during this process is use a little bit of NADH, or NAD plus, right? And use a little bit of energy from this process, from acetyl-CoA. So it's a little bit less energized because we've taken that electron that represents that energy and tacked it onto NADH, which is also going to go on later to the electron transport chain. Okay, so now that we have our main reactant in the citric acid cycle, or the Krebs cycle, we'll go ahead and start our cycle. So I'm going to draw a circle for you with a couple arrows indicating that this is a cycle. All right, and throughout this process, we have several reactants and products. All right, so one of those products, or one of those reactants, is ADP plus P. We're going to yield ATP from this one. Okay, we're taking a little bit of energy from this acetyl-CoA. We're also going to gain a little bit of energy in the carried on NADH. So we're going to convert our energy carrier, NAD plus, to NADH by attaching that electron. And furthermore, we are also going to generate a little bit of FADH as well. All right, so we're ultimately yielding 3 different forms of energy carriers, 3 different energy carriers to be used to donate electrons to our electron transport chain. Also, as a result of this Krebs cycle, we're going to produce more carbon dioxide, which I'm going to draw an arrow up here. Okay, so 2 times during this process, we are releasing carbon dioxide, which is going to be released out of the cell. as a waste product. All right, so that kind of wraps up the Krebs cycle. In terms of our ATP count, here we are generating a little more ATP, 2 molecules to be exact, so I'll tack that on to our ATP count. So far we've yielded 4 ATP, which isn't a whole lot. But remember, the whole point of this process is to generate enough electron carriers to be passed on to the electron transport chain to donate electrons, all right, in order to create lots and lots of ATP. So we're going to zoom in on the electron transport chain. Actually, before I do that, I want to indicate our electron carrier, or our energy carriers, and bring them towards the electron transport chain. So first we're starting with NAD and NAD+, or NADH, sorry, is coming to the electron transport chain. We also have, from the Krebs cycle, we have NADH coming from our oxidation of pyruvate. We have NADH also coming from the cycle itself, as well as our FADH, which is carrying energy, electrons from our FAD, or from our Krebs cycle. So let's go ahead and zoom in on our electron transport chain. All right. And our electron transport chain has, I'm going to erase some space for our 1, 2, 3, 4, 5. Okay, so these spaces that I just erased are going to represent areas for our the parts of the electron transport chain. These 4 electron carriers, these are proteins, transmembrane proteins, and I'm also going to draw our ATP here, synthase here, okay? So just to get our bearings, remember this part is the matrix. This is our membrane, our inner membrane. We have our intermembrane space, and this is the cytosol of our cell, okay? I'm going to erase this intermembrane space just so it doesn't get in the way, but just so you have your bearings, make sure you know where you are when we start this process. And what's happening here is we're going to have all of our energy carriers, NADH and FADH, that were generated during the Krebs cycle, and the glycolysis that are going to donate electrons to our electron carriers, our electron transport chain. Okay, so our NADH is going to donate electrons here, and so we're going to yield NAD+. Our FADH is going to donate electrons and yield FAD+. All right, so these are our products and our reactants, but as they donate an electron, all right, during this process, the electron is going to go across the electron transport chain. Okay, so we're adding these electrons to this chain, they're being carried across, and every time they are carried across, we are going to also pump a hydrogen. All right, through each one of these processes, every time an electron zips through, it pumps 1 hydrogen across into the intermembrane space. And remember that these NADH to NAD+, this hydrogen is popping off. That's where the hydrogen's coming from. Okay, so the hydrogen's popping off, and then we're able to pump it across the membrane into that intermembrane space. And when we pump these hydrogen ions, this is creating a concentration gradient. So if we were to draw a little arrow of like low concentration to high concentration, this is low concentration, high concentration. We are moving from an area of low to high, which goes against the concentration gradient. So we have a really high concentration of these H+ molecules, so they're going to want to get out in any way that they can, okay? Before they can get out, we're going to talk about what's happening to the electrons, however. And remember, these electrons, and these are going to be added to molecular oxygen, which is going to split in half and take up hydrogen and form water. Okay, so I'm going to draw that happening. All right, so we also have O2, which is present in the matrix. It's going to grab onto this electron. This is going to be used to split oxygen into just 1 molecule, so that we end up with H2O. And I'm going to just draw our hydrogens popping on there. Okay, so we have our oxygen and our hydrogens. Now you might also be wondering where are these hydrogens coming from. I thought they were on this side, in the intermembrane space, but we are going to use ATP synthase to allow those hydrogens to come back, okay? So through passive transport, all right, hydrogen ions or protons are going to pump across this ATP synthase, all right? So they're going to, and like I said, passive transport, they're not being pumped, sorry. They're passing through this protein, this ATP synthase, and every time we have a hydrogen passing through, ATP synthase is going to take ADP, which is already present and bound to the protein, and it's going to take P. And as we use this energy, or as ATP uses some of the energy from this hydrogen ion, as it gained a lot of energy being in this little space, it's going to pop those two together. I'm going to erase this. And we are going to generate ATP here. And because for every hydrogen we get one ATP, it's going to generate almost 80% of ATP made during breakdown of glucose. Okay, so we're actually going to get 32 ATP from this process. I'm going to put that onto my count. 32 ATP. The electron transport chain. All right, so we're generating a lot of ATP in this process where we're using energy from a proton gradient in order to generate ATP. This is also called chemiosmosis. We'll hear it referred to as chemiosmosis, okay? Using a proton gradient to form ATP. Now, let's zoom out again, if I can. There we go. All right, so now we have the full picture, and I know it's kind of small in some areas, but we've got our whole cycle of glycolysis donating energy through energy carriers to the Krebs cycle, where we have pyruvate being oxidized, producing more energy carriers passed on to the electron transport chain. We use that energy in the form of electrons, pass them across a couple proteins, which pump hydrogen protons across the membrane, and we use those protons to generate ATP in the end, all right? So we've produced a lot of ATP through this process, as well as carbon dioxide. We have produced water, all right? So when we're thinking of our products and reactants, I'm gonna write out our formula up top. So we start out with C6H12O6, which is also known as glucose. All right, plus 6O2, right? We're using our oxygen over here. Plus 6O2 yields 6CO2 plus 6H2O plus ATP. All right, and what I'd like you to do as I stop the video, go ahead and figure out where are these entering, where are these products being released, and why are they important. And if you are to continue on after you feel comfortable with this process, I would encourage you to do the case studies, because they show you if we inhibit one or more of these processes, if we can inhibit the uptake of one of these processes, how does that affect diseases? If we inhibit the electron transport chain, what diseases can result? And these are the types of examples that you'll see on an FRQ, both in the test and in the exam. So I would encourage you to do that. It really tests your understanding instead of just being able to regurgitate this process, okay? So I would encourage you to ask for help if you need it. Otherwise, happy studying.